I think that when Pisan thought about canSAR, we were starting to feel a bit overwhelmed by the amount of data that's available, right? And it's very multidisciplinary and it's really broad. And when you're working in multidisciplinary research and the initial aim was drug discovery, and you have to decide which target you choose to design the new cancer drug, you have to take all these data into consideration, and there's increasingly researchers are putting data out there, but it's not always easy to put everything together and analyze it all together because it belongs to very different disciplines, and that was a real challenge. So that's kind of the vision of canSAR, and why we started building it. canSAR is a multidisciplinary research that brings data together and is designed to be searched via different ways. If you type PARP1, which is a protein that's involved in cancer, you can see here several results pop up. This landing page is a bit of an overview of all the information available. We have different data types that you can click and navigate through. You can dig down into the data. You can see the approved drugs, the compounds in clinical trials, the mutation, expression over time, copy number variation, and the cell lines that are more or less affected. We have also the chemistry wheel as well, where we show all the data that we have of the small molecules binding. This is a cellular localization. The cancer type section of the wheel tells us which cancer types we predict that are going to be more affected by this protein. The signaling section, where we have our own curated version of the interactome. You can see the proteins that are known to interact with PARP1, and you can also see the evidence of these interactions. Predicting druggability or ligandability is one of the strengths of canSAR. I think we are the largest public resource that predicts druggability, and we do this across all the proteome, not just for cancer. We've developed a machine learning artificial intelligence model to predict druggability. And here we have the 3D structure of this chain, and here you can see this is the cavity that we are predicting to be druggable. And we can zoom in and see how this ligand, for example, is interacting. So this happens with all the sections of the wheel. We can always go on top and go back to the molecular synopsis to explore the other areas. But the idea is that you have in this wheel a first glimpse of the data that's available, and then you can deep down into more defined data.